Hi, welcome everyone. This is Irene, Product Manager of Molecular Biology at GenScript. And in this session, we will invite Dr. Jiangbing Zhou from Yale University to give us a talk about polymeric nanoparticles for non-viral gene delivery. There will be a Q&A session after the talk, so please type your questions in the chat box, and we will answer them in the order it was received. And in case there are too many questions, we will just forward the answer to you offline. Dr. Jiangbing Zhou is Associate Professor of Neurosurgery and of Biomedical Engineering at Yale University. And his research mainly focuses on the development of technologies for non-viral gene delivery and drug delivery to the brain, as well as an application of these technologies for treatment of neurological diseases. So let's welcome Dr. Zhou to give us talk on the non-viral gene delivery.
Great. Thanks, Irene, for your introduction and for the invitation. It's a great pleasure to speak here. For those who don't know me, my name is Jiangbing Zhou. I'm an associate professor of neurosurgery and now biomedical engineer at Yale. So today I will talk about the development of polymeric nanoparticles for non-viral gene delivery. So I think for this audience, probably I don't need to talk too much about what's gene therapy. So gene therapy basically can be defined as a group of techniques that can be used for correct defect genes responsible for disease development. So there are only three different types of gene therapy approaches, including gene augmentation or gene replacement therapy. So typically, one needs to deliver either cDNA or mRNA into target cells. The second type is called gene suppression. So in this case, either siRNA, sRNA, or SOs can be delivered to target cells to suppress target gene. So the last one is called genome editing. So in this case, one can deliver some proteins like CRISPR to cells to edit the cell genome. So gene therapy actually is not a new concept. The first clinical trial can be dated back to 1989. However, it's also recognized gene therapy is extremely challenging. So the major setback actually happened in 1999. So when a boy received gene therapy treatment by infusion of adenovirus, unfortunately the boy died from this viral infusion. So this caused tremendous caution by the FDA. So after that, so a lot of regulation were developed. So as a result, it's very difficult to translate gene therapy into a clinic. So as shown here, the first gene therapy product was not approved in this country until 2017. Fortunately, in the last few years, gene therapy field has seen tremendous progress. By now, there's over 8 different products which have been approved by the FDA for clinical use. So if we look into current clinical trials about gene therapy, you may see that, as shown here, most gene therapy are developed to tackle monogenic disease. I think that make a lot of sense. So you just need to modulate one single gene. So if you look into the delivery system, you may notice that majority of the clinical trials are using viral vectors, as shown here, retrovirus, AAVs, adenovirus, so on and so forth. So clearly you can see that viral vectors are dominantly used at this moment, particularly AAV virus, attracting significant attention over the past many years. So of course, the viral vectors are used for reasons. The major reason is that the viral vectors can transduce target cells in high efficiency. So this viral vector is selected because they have been evolved to have high affinity with cells. You can hijack the human cellular system, for their own genome production. However, it's also very recognized the viral vectors have a lot of problems. For example, they have safety concerns, potentially generate immunogenicity. For example, for certain viruses, you cannot deliver a virus for more than one time. They're also very difficult to produce. Actually, right now, this has been a major hurdle for clinical translation of many viral vector-based gene therapy. And also, there's some limitation about loading capacity, particularly for AAV. So it's well known that typical one AAV vector can only accommodate a transgene in size less than 4.7 kilobase pair. Because of this concern, there's a lot of efforts made over the past decades, about development of next generation vectors for gene delivery. So among them, non-viral vectors are very appealing. So it is considered as probably the safer alternative. So one of them is based on nanoparticles, synthetic nanoparticles. Of course, what are nanoparticles, right? So nanoparticles defined as particles in size about 20 to 500 nanometer. So basically that device in nano size. So when utilized for gene therapy, typically nanoparticles refer to those particles in size about virus. As shown here, AAV typically about 20 nanometer, and then largest virus used for gene delivery is adenovirus, and the lentivirus typically about 100 nanometer. So in this field, a lot of people just make synthetic particles, or another way is that synthetic artificial virus to mimic these nanoparticles. So we understand is that it's proposed actually that nanoparticles have a lot of advantage, just based on the chemistry. For example, they're pretty safe, because they're all made of synthetic materials. They're relatively easy to produce. They have tons of flexibility in terms of surface chemistry and for targeting. And also they're capable of delivering large payloads. Actually, there's no limitation for most nanoparticles for gene delivery in terms of payload size. The disadvantage has been also obvious. Actually, it's not considered. Nanoparticles have very limited efficiency. So it's always thinking nanoparticles may not be able to use for gene therapy, at least in short time. However, this concept was dramatically changed over the past few years. So we know that COVID-19 started probably just a few years ago. Started 3 years ago actually. So after that, many companies started to develop gene therapy vaccine. Actually, shown here, the traditional based vaccine like protein based or inactive viral based vaccine products. But to the end, the real winners are lipid based nanoparticles. So we know that most people in this country actually received LNP-based vaccine. So this really, generated a lot of excitement in the field. So by now we recognize LNP-based gene therapy has a lot of advantage. For example, it has been proved to be safe. This particle can be utilized to deliver gene therapy high efficiency. So of course, there's many challenges identified. For example, it's well known that most LNPs are somewhat unstable. They cannot produce prolonged gene expression. And also in terms of production, cost is also pretty high. So actually in the non-viral field, other than LNPs, there's another type of gene delivery system called polymer-based gene delivery system. So in this case, polymer can be utilized to condense genetic materials into polyplex nanoparticles, or polymers can be utilized to synthesize solid nanoparticles. So basically like a solid ball, and then encapsulate whatever genetic payload. So when we're talking about polymer-based gene therapy, probably in most people's mind is this first generation polymers, including PL, PEI, or PBAEs. Particularly, most people probably know PEI. Actually, PEI still being used in current clinic for gene therapy. So it's most used for in vivo, ex vivo use. So the PEI-based, actually the PEI, first generation based polymers, they can deliver gene quite high efficiency. But the delivery mechanism is quite simple and straightforward. Essentially, everything is based on charge. For example, because of charge, this polymer can condense genetic payload into nanoparticles. And also because of the positive charge, these particles can cross cell membrane and escape endosome. So however, this charge also creates a lot of problems because it's a non-specific charge. They are very toxic to human cells and the physiological system. So as a result, most first generation polymers actually cannot be used for in vivo gene therapy. So many years ago, when we started work on developing next generation polymer for gene therapy, we think what we should do. So we start with thinking about or examining what's cellular mechanism for gene delivery. So as shown in this cartoon, so for whatever vector you use, the first step always cellular uptake. So your carrier needs to get into cells. So typically through endocytosis pathway, so which means that nanoparticles will get into endosome. The second major step is that your nanoparticles need to escape endosome. Otherwise, the particle will be transported to lysosome, so which have a very highly acidic environment. So essentially, most materials cannot survive. So to avoid lysosome, the particles need to escape endosome. So the second major step is called endosomal escape. So after that, your payload need to be released. And if DNA is payload, then your DNA need to transport into the nuclei. And to the end, the protein product can be produced. So molecular mechanism level, actually the mechanism is pretty clear. So we then see whether this mechanism really is, for example, if we achieve this mechanism, and can we really deliver gene well? To test this, now we use very common use materials called PLGA, polylactic galactic acid. So PLGA actually one of most often used materials in current clinic. So this materials was approved by the FDA in 1969, and then right now used in many clinical products, mainly because they are highly biocompatible. Imagine they degrade into lactic acid and glycolic acid, both exist in human body. So it's widely used, but it also knows that this materials cannot be used for gene delivery well, mainly because they are electrically charged. For example, when we test the use of PLGA particles for gene delivery, we can see that efficiency-wise, probably 6 log below than lipofectomy. Lipofectomy, everyone knows that it is the leading commercial agent. So PLGA particles themselves cannot be used for gene delivery. So then, we just keep adding modality into the PLGA nanoparticles, as I just mentioned, right? 3 major steps: cellular uptake, endosomal escape, and then in this case, we deliver DNA. We need to facilitate DNA transport into the nuclei. So without going into details, I just show you the results and the finding we have. So here, for example, we can enhance cellular uptake by adding the cell penetration peptide on the surface. We can improve the nuclear transport by include a small molecule which can transiently open the nuclear pore complex. We can also include a molecule called CQ, which can facilitate endosomal escape. So we can see that if we add 3 modules onto the PLGA nanoparticles, the resulting particles can deliver gene in pretty high efficiency, right, close to lipofectomy. So we can further add another peptide to the surface. So in this case, the second peptide is also designed for cell penetration and endosomal escape. As you can find here, right, after a very complicated chemistry, we end with nanoparticles, several different types of nanoparticles, which can deliver gene very well. in terms of efficiency, they are comparable to lipofectomy. So, even after this such a complex chemistry, they still with sphere shape, about 100 nanometer in size. And then they can deliver, this is the surface just for model gene. And then we testing these particles for deliver gene therapy. So in this case, we choose PLK1 sRNA. So remember probably 15 years ago, sRNA is the hottest area in this field. So that's why we choose to deliver sRNA for gene therapy in vitro and in vivo. So as shown here, this is a cell culture work. We demonstrate that treatment with this SDI-PLK loaded nanoparticles can significantly enhance cell penetration by over 90%. And then we test these particles for in vivo gene delivery. So in this case, we add one more modality onto surface nanoparticles, which is peptide RGD. So RGD is designed for tumor targeting, for them as shown here. Sorry, I didn't include that here. So anyway, so this is this modality was added to improve tumor targeted delivery. So as a result, we found that these particles really, after intravenous injection, can suppress tumor development. We found that these particles works to inhibit PLK1 expression. Notice is H1E and tunnel staining. So basically, we demonstrate that now we can start with PLG nanoparticles, which cannot deliver gene by themselves, and then we can add a modality to those particles to achieve cell uptake in the soma escape, and also nuclear transport. We can obtain nanoparticles which can deliver gene very well. So the question is that really such complex nanoparticles are answer a lot. So actually it's pretty clear. It's not the answer because it's so complicated. It has zero possibility to be translated in clinic. However, this experience really give us a lot of inspiration for development of next generation polymers. So in this case, we developed a new chemistry for polymerization of 3 molecules into one single polymer, polyamine coester. So this chemistry is unique because we can control charge, which is provided by the tertiary amino group, and the molecular weight, of course, by reaction time, and the hydrophobicity, which is provided by the lactone group. As a result, we can obtain a group of polymers. As shown here, we can get liquid polymers or solid polymers simply by adjust the amount of lactone group or size of lactone group used for polymerization. So we tested these polymers both liquid and solid. We found that this liquid polymers can be utilized for gene delivery very well, as shown in this figure. So we found that these liquid polymers can condense DNA into nanoparticles, about 100 nanometer. In terms of efficiency, is about 10 to 50 times greater than lipofectomy, the needed commercial agent. The problem is that we found this lipid, this is a polyplex, similar to the lipid nanoparticles. This cannot be offline because they are unstable in structure. Of course, we also testing this solid polymer in this family. We know this solid polymer can form solid nanoparticles, which are very stable. But unfortunately, solid polymers in this family cannot deliver gene well, as shown here, right? The several log behind, below commercial agents. So over the past few years, we have been working on development next generation polymers for gene delivery. So again, without going into details, so we extensively change the chemistry and then try many different combinations. We have screened probably over 300 different polymers, And we, by far, we have identified several polymers which can deliver gene very well. For example, one lead polymer called P60OD, which can form particles, and show here, nice spherical shape. We found this polymer can form particles which can deliver gene very well. Either DNA or mRNA. In terms of efficiency, is probably 10 to 50 times greater than a commercial agent. So as I mentioned, our goal is that we want to obtain nanoparticles which are highly stable, and can also, in addition to having high gene delivery efficiency. So we've demonstrated that indeed these particles are very stable, and after synthesis using microfluidic system, this particle can be lyophilized into powder, as shown here. So whenever you need to use these particles, you can simply reconstitute your particles with saline or cell culture media. We found that this reconstitution doesn't reduce delivery efficiency. We also found that these particles are quite unique. And remember, I mentioned one disadvantage of LNPs is that they cannot deliver gene over a long time. So different from lipid system, these particles are solid and they release the payload over time. So we found that one single treatment, in vitro study, can produce gene expression over 2 weeks. Of course, here I only show 1 week data. So actually, one single treatment can produce sustained gene expression. And also some other very unique properties. For example, we tested one of the lead particles, P60OD MPs. We found those particles can transfer to the lung very well after intravenous injection. So probably you know that most particles after intravenous injection should land into the liver and transfer to the liver. And for example, here is MSC3 LNPs, with alignment proprietary LNPs. So we can find that after our injection, the particle transfer to the liver. But our particles, have high specificity with the lung, can transfer to the lung very well. And also when the genome editing machinery was delivered, this particle can edit the lung cells with high efficiency. So compared to other systems, our solid polymer-based nanoparticles have several advantages. For example, we demonstrate our particles can deliver gene in high efficiency. We compare our particles to many different systems, including LNPs, alignin-based LNP or Moderna-based LNP. In all cases, we found our particles are more efficient than this commercial available systems. Of course, our system is very simple, just one single polymer, and our particle can produce sustained gene expression. And also, our system have major advantage in terms of stability. We can lyophilize the particles, and the particle can be utilized for many months and years. And also, production is relatively simple and straightforward. Can be done using a traditional emulsion approach or microfluidic system. So acknowledgement, of course, I need to thank the lab. A lot of appreciation to my lab members, either current lab members or previous members. I also thank my collaborators, including Yong Hui, James Hansen, Mark Lier, and Bob Casterson at UAB. And of course, I would like to thank my funding agency, including NIH, Gilbert Foundation, M2 Foundation, and patrons. Thank you for your attention. I'm more than happy to take questions.